Hi biology students, this is Mrs. Miller again, and today we're going to wrap up with our last section of molecules of life. So section 3 is going to cover something called enzymes. You probably won't notice it happening, but right now your body's going through a series of reactions that allow us to live. Without certain things within our body, these reactions would be really slow, and then we wouldn't be able to survive very long. So what we're going to look at today are enzymes, and before we get to that, we really need to focus on something called activation energy. So many reactions within our body require energy to kickstart that reaction. That's very normal. We're going to see some things like maybe as you go into chemistry, we have to add flames to something or increase the temperature or drop the pH or something like that to make a reaction actually happen. This is called activation energy. What I can see here is the amount of activation energy. So I need to get all the way up to the top of the hill before this reaction can take place. Over here, I also have a little hill that I need to get over, but it's much less. So these activation energies within our body might be very high, making it difficult for some of these reactions to take place. Some of the reactions within our body are too slow or have really high activation energies. In fact, if we think about how long it takes us to digest food, if we didn't have some of the things that we have within our body, the activation energy on that is so slow that if you ate a meal today, you'd actually starve to death because the amount of energy that it would take to get over that activation energy and start you digesting is simply too high. Thankfully, we have certain reactions that are made possible with something called a catalyst. A catalyst is a substance that speeds up the rate of reaction, and this is just another name for an enzyme. What a catalyst does is going to allow us to lower the activation energy. So here I can see that hill that I have to climb right here is really steep before I can get down. So let's say that this is Granite Peak. I make you guys walk up Granite Peak. I know that would be really mean, but finally you get there and you can ski down. With an enzyme, what we're doing is we're making this easier. So now, all you have to do is get yourself to the chairlift. It'll bring you up there, so a lot less energy needed to get on that chairlift, and then whoop, you can go down the hill. So it's a lot easier to do things with an enzyme versus without because that activation energy is so much lower. Much of what you see on this particular slide is something that we've already talked about on the previous slide, but I do want to point out something with enzymes. Enzymes are going to be special types of proteins. So that's an important thing to keep in your mind. In the last unit, we talked about proteins. So remember, proteins help with structure, they regulate a lot of things, and one of the things that they regulate within our bodies is the rate of reactions. So it's these specialized proteins that act as biological catalysts. They help to speed up reactions by lowering the activation energy, and it makes it easier or takes less time for that reaction to take place. So here I have a picture of an enzyme, and then we're going to be working with something called a substrate. A substrate is just the thing we want to break down or build up with the help of an enzyme. One real-life application that we can look at with enzymes is looking at the amount of carbon dioxide within our blood. So how big of an effect do enzymes actually have on reactions? Well, within our body, we are constantly going through some changes, and that would be where we're producing carbon dioxide and water as we go through a process called respiration. What that releases is something called carbonic acid. And this whole process is really slow without enzymes. So what happens is CO2 begins to build up in our body faster than our bloodstream can remove it. If we didn't have an enzyme, that would be very dangerous for us. Thankfully, blood has an enzyme called carbonic anhydrase that speeds up this reaction and gets rid of this carbonic acid within our blood. Without this enzyme, your blood would essentially poison you because the pH of your blood would start to drop very quickly. And we know that if our blood pH drops quickly, we can go into a coma and we can die. So we really do need these enzymes. One thing I want you guys to know about enzymes is that they are super, super specific, and they can actually only be used by one chemical reaction. So over here, I see 3 types of enzymes, enzyme 1, 2, and 3. Here, I can see that these 2 circles will fit perfectly into this spot. In enzyme 2, it's this triangle that fits, and in enzyme 3, we have this other shape that fits. If I try to take this triangle and put it into enzyme 1, it simply wouldn't fit. So that wouldn't work for that particular reaction. Another important thing to notice is that enzyme names usually describe the reaction it catalyzes or helps to go through. For example, lactase is a common enzyme. Lactase breaks down lactose in dairy products. So notice it starts with the same and it just changes the ending to ase. Ase usually tells us that we're dealing with an enzyme. So if you are lactose intolerant, you have no enzyme to break down dairy products. So you might take lactase enzyme in the form of a pill so that you can actually have those dairy products. The next thing that we're going to look at is something called the enzyme substrate complex. So enzymes have a site where reactants are brought together to react. Again, each protein has a very specific shape, or each enzyme has a very specific shape that allows only one type of substrate to bind with it. The substrate, or the thing that we want to change, is going to bind to a site on the enzyme called the active site. So this yellow thing here is the enzyme. The actual site that's specifically shaped, that's called the active site. And then in here, the thing that's going to come in is called the substrate. When the 2 of those things bind together, we call that the enzyme substrate complex. What I really want you to know is that substrates and active sites fit together like a lock and a key. So I carry around a lot of keys. For example, I have a house key, I have my car keys, I have my classroom keys. If I tried to use my classroom key in my car, it's not going to work. So it's just like if I put the wrong key in the lock with my enzymes, it's not going to work. So you have to have that proper match for the enzyme to go through that reaction. What we're going to see is that the enzymes and the substrate are going to be held together until our reaction is completed. Products are released and the enzyme is going to start this process again. So nothing happens to our enzyme. It gets used over and over and over again. What we see is we have a very specific shape with our active site on our enzyme, and the substrate is going to pop in. When that happens, it's like the enzyme changes shape just a little bit to bind them together. Then what happens is we're going to change this product, or change this thing into a new product. So in this case, it's breaking it down. And then the enzyme is going to release those products to go out. But what's going to actually be happening then is our enzyme is going to go back and it's going to start over again. So the enzyme can go through this over and over and over, and it always just has to meet up with the same substrate and then produce those same products. Another important thing to understand is the fact that enzymes are affected by outside factors. 2 of the major factors that impact them are things like pH and temperature changes. So enzymes within our body tend to work best at 37 degrees Celsius, which is our normal body temperature, which is about 98.6 degrees Fahrenheit. If we get too cold or we get too hot, our enzymes change within our body and we can't use them. So if an enzyme is brought out of its comfort zone, it becomes denatured. This means it changes the shape so it won't work anymore. It might work again once the temperature comes back down, but it might not. So here I can see 3 different enzymes. And what I'm noticing is that at the highest point here for red, it's about at a pH of 4. If all of a sudden this enzyme was put into something like a pH of 8, it's not going to work anymore. Just like our green one, I can see that it works best at about a pH of 9. So if I put it at a pH of 6, it's not going to work very well anymore. So when we change the pH or we change those temperatures, those enzymes won't work well anymore because they become denatured and they change their shape so they can't work. Then to wrap up, cells can regulate enzymes in many ways. First, proteins can turn enzymes on or off at any time that they need them. So certain enzymes might be working during part of the day. They might not work after you eat, or they might work really, really hard after you eat and things like that. But the roles of enzymes, of course, are to regulate chemical pathways. So we want to make sure that your body is in a nice state of homeostasis or a stable internal conditions. They might be making materials that cells desperately need. So as we eat, we get our food that gets broken down into different types of sugars. So we need our enzymes to help us with that. Enzymes help to release energy. So as they break bonds, they release that energy. And of course, they're also going to be transferring information. So hopefully you guys have a better understanding of enzymes now. If you have any questions, please let us know. Otherwise, have a wonderful day.